Dr. Sage here, back with the third of 3 video lectures on phylogenies and the history of life. By the end of this video, you should be able to describe horizontal gene transfer, illustrate how eukaryotes and prokaryotes transfer genes horizontally, and understand why horizontal gene transfer is problematic in creating phylogenetic trees. So the concept of phylogenetic trees began with Charles Darwin. Since then, new technologies and techniques have changed our classical view of evolution. We used to think that fungi and plants were closely related. However, now we know that fungi are actually more closely related to animals than they are to plants. That mushroom is more similar to you than that mushroom is to a plant. We used to think that birds were a separate class of vertebrates. Now we know they are descendants from the theropod dinosaurs. Okay, so the dinosaurs didn't actually die out. Some of them evolved into birds. Dinosaurs, the crocs, which is the alligators and the crocodiles, and birds are more closely related to each other than dinosaurs and crocs are to modern-day reptiles. So in an earlier lecture, we discussed how we define a species. The main way we define a species is known as the biological species concept. We define a species as organisms that in nature can breed with each other. They can successfully mate and produce viable and fertile offspring. However, that's not the only way to define a species. And that definition, the biological species concept, doesn't work for all organisms. For example, how do you determine if a species of bacteria is the same as another bacteria when bacteria don't mate with each other? So you can't see if they can mate and produce viable and fertile offspring since they don't mate. They reproduce asexually. So we also have the phylogenetic species concept. In this, a species is a population or set of populations characterized by one or more shared derived characters. Now, which concept you use depends on what research you're doing. So if you're using an organism that does mate, then you can most of the time use a biological species concept. If you're using an organism that mates asexually, or an organism that's extinct, so you can't tell if fossils can mate with each other, you can't use a biological species concept. In an earlier course, you learned about genetics, and you learned about the work of Gregor Mendel, and Mendelian inheritance, Mendelian genetics. But Mendelian inheritance and its modifications assume that genes are transferred from the parents to the offspring. That's known as vertical gene transfer. Genes are going from the parents to the offspring, to the grandkids, etc. And that mutations are the primary source of new genetic information. However, this is not the complete story in prokaryotes and even in some eukaryotes, because there's also something called horizontal gene transfer. So instead of genes being transferred vertically from one generation to the next, they can be transferred horizontally. That's transfer of genetic material between unrelated species. So horizontal gene transfer, transfer of genetic material from one species to another species. This occurs more often in prokaryotes than eukaryotes. Mutations and horizontal gene transfer are important sources of genetic variation. 3 primary modes of horizontal gene transfer are transformation. That's where naked DNA is taken up by bacteria. So for example, if you have a bacteria here, this bacteria has a chromosome. But if there's a piece of DNA outside the bacteria, that piece of DNA can go into the bacteria, and that genetic sequence can then be integrated into its chromosome. That's a process called transformation. Then we have transduction. Transduction is when genes are transferred via a virus. So again, if we're looking at bacteria that has its chromosome, there are viruses that infect bacteria. They're called bacteriophages. The bacteriophage can insert its genetic material, its DNA, into the bacteria, and then that DNA can be integrated into the chromosome of the bacteria. That's called transduction. Or you can have conjugation. Conjugation is when genes are transferred between bacteria via a pilus. So that's basically like a tube that connects these 2 bacteria, and then this bacteria can transfer this genetic information, this DNA, to this other bacteria. That's called conjugation. We'll probably learn more details about that when we learn about bacteria. Horizontal gene transfer occurs more often in prokaryotes, but it can also occur in eukaryotes. One common way this can happen is using things called transposons. Okay, you can also think of them as jumping genes. What a transposon or transposable element is a chunk of DNA, a piece of DNA, a sequence of DNA that actually has the ability to cut itself out of the chromosome and reinsert itself somewhere else. And that can also be between different species. So eukaryotes can have transposons. So information can move from one species to another. Another way that information moved from one species to another happened in the early evolution of eukaryotes. So prokaryotes evolved first. So you have a prokaryotic cell. Recall that prokaryotes don't have any organelles, no membrane-bound compartments inside the cell, but they do have a plasma membrane. Prokaryotes also have a nucleoid. That's the region inside the cell where you find their chromosome. Now, in evolution, something that happened is that the plasma membrane started to fold in. Okay, so now you have some membrane inside the cell, but it's actually part of the plasma membrane. It's just folding in on itself. And you can see that in modern-day prokaryotes. There's many prokaryotes that have the plasma membrane invaginating or folding into the cell. If this plasma membrane started to wrap around the nucleoid, and if this membrane got separated from the actual plasma membrane, then that's how you can start to form the first organelle. Okay, in this case, the nucleus, because now you have the membrane surrounding the chromosome. And that's how you could get some of the first eukaryotic cells forming from prokaryotic cells. In those early eukaryotic cells, they didn't have all the organelles that modern eukaryotic cells have. And a couple of the organelles arose through a method that's different from how we got the rest of the organelles. That would be the mitochondria and the chloroplast that you should have learned about in Bio 1. What happened with the mitochondria and the chloroplast is far back in evolutionary history, there was an early eukaryotic cell, and that eukaryotic cell went and it ate a prokaryotic cell. Okay, eukaryotic cells eat prokaryotic cells all the time. You can watch that happen. Okay, but instead of digesting that prokaryotic cell, what happened was that prokaryotic cell started living inside the eukaryotic cell. That's called symbiosis. You can also observe that happen. Prokaryotic cells living inside eukaryotic cells. Those cells living symbiotically, it was an advantage for both cells. Okay, that prokaryotic cell was now protected because it was living inside the eukaryotic cell, so it was less likely to actually get eaten by another organism. And the eukaryotic cell had an advantage because that prokaryotic cell, let's say it was a prokaryotic cell that could do photosynthesis. So plants are not the only thing that can do photosynthesis. There are bacteria called cyanobacteria that can do photosynthesis. What that means is that bacteria is producing glucose, which some of that glucose can then go to that eukaryotic cell. So they were living symbiotically. They both had an advantage living together. Over the evolutionary time scale, that prokaryotic cell lost its individuality and it just became part of the eukaryotic cell. And that's where the chloroplast came from. The chloroplast used to be a separate living organism. It used to be a prokaryotic cell. That's also how we got the mitochondria. There was a prokaryotic cell that could do cellular respiration. Okay, a eukaryotic cell went to eat it, and then they started living together symbiotically, and it became part of the eukaryotic cell over the evolutionary time scale, and that's where the mitochondria came from. This is called the theory of endosymbiosis. Okay, now, when you first hear that, that might sound like very science fiction-y or like I'm feeding you a line of bullshit. Okay, but there's actually a lot of evidence supporting this. Okay, for example, the inner membranes of the mitochondria and the chloroplast are similar to the plasma membranes of prokaryotes. Another example, you have all of your genetic information, all of your chromosomes inside your nucleus, because that's where you store your chromosomes. Except you have one more chromosome somewhere else. You have one chromosome inside your mitochondria. Why? Because the mitochondria used to be a separate living organism. It used to be a prokaryotic cell that had its own chromosome. So you have 46 chromosomes inside your nucleus and one chromosome inside the mitochondria. Now, if you look at those 46 chromosomes inside your nucleus, they look like eukaryotic chromosomes, which are like long and linear chromosomes. That's what eukaryotic chromosomes look like. You're a eukaryote, so it makes sense that your chromosomes are eukaryotic chromosomes. However, if you look at your chromosome inside your mitochondria, it doesn't look like this. It's small and circular, which is what bacteria, prokaryotic chromosomes look like, which makes sense because the mitochondria used to be a prokaryote, a separate living organism. You also have ribosomes. Okay, your ribosomes are floating around in your cytoplasm and stuck on the outside of your rough ER. Except you have ribosomes somewhere else. You also have ribosomes inside your mitochondria. Why? Because the mitochondria used to be a separate living organism or prokaryotic cell that had its own ribosomes. So that's why the mitochondria still has its own ribosomes. Now, eukaryotic ribosomes and prokaryotic ribosomes, they're very similar to each other, but they're built out of slightly different pieces. Well, the ribosomes float around in the cytoplasm, stuck on the outside of the rough ER, they're eukaryotic ribosomes, which makes sense because you're a eukaryote. But the ribosomes inside your mitochondria, they look like prokaryotic ribosomes. Why? Because the mitochondria used to be a prokaryote. More evidence. When do you get new organelles? Okay, when does that happen? When your cell divides. You have one cell, it's going to become two cells. Well, each of those two cells, they need a nucleus. So you go from one nucleus. When do you get two nuclei for the two cells? When that cell is dividing. Okay. When do you get more ER? When do you get another Golgi apparatus? When does that happen? When the cell divides. When does the mitochondria divide? Whenever in the hell it wants. Why? Because it used to be a separate living organism that would divide whenever it wanted. Okay, everything I said about the mitochondria, the same is true of the chloroplast. Okay, they have their own ribosomes, they look like prokaryotic ribosomes. They have their own chromosome, it looks like a prokaryotic chromosome. They divide whenever they want, that's why plants have more than one chloroplast inside each cell. The mitochondria and the chloroplast used to be a separate living organism, it used to be prokaryotic cells. What that also means is some prokaryotic genes have now been transferred to a eukaryotic cell. Okay, so that's another example of horizontal gene transfer. Genes being transferred from one species to another because we now have prokaryotic genes inside our eukaryotic cells. So horizontal gene transfer has been proposed to be important in the evolution of prokaryotes. And it may lead to an alternative to the tree of life model of evolution. Instead, this would be called the web of life model of early prokaryotes, and it may be more appropriate. Horizontal gene transfer mechanisms evolve such that eukaryotes evolve from a pool of prokaryotic species, some giving rise to mitochondria and some to chloroplasts. In other words, instead of a standard tree of life where there's one tree trunk, there's multiple trunks here, and they're transferring genes between each other, okay? Including transferring from prokaryotes, like the cyanobacteria, becoming the chloroplast now inside eukaryotes, or the proteobacteria now becoming the mitochondria inside eukaryotes. So this horizontal gene transfer creates more of a web of life than a tree of life. Another alternative is the ring of life model, proposed that all three domains evolve from a pool of prokaryotes swapping genes via horizontal gene transfer. This may help explain how certain eukaryotic genes more resemble those of bacteria, while others resemble those of the archaea. Only more research will distinguish between these different models of evolution. So this concludes a set of lectures on evolution. We went through several chapters learning about the basics of evolution. For the remainder of this semester after this, we will be learning about the different types of organisms living on this planet. Okay, we're going to start learning about the prokaryotes, the single-celled organisms, bacteria, and archaea. And we'll also learn a little bit about the abiotic viruses, so non-living, but they infect living things. Then after that, we'll learn about the eukaryotes and the different kingdoms of eukaryotes, things you're familiar with like plants, animals, fungi, and something you're probably not very familiar with, the protists. So the rest of the semester we're going to spend learning about all of the different types of organisms living on our planet. So until then, this has been Dr. Sage.